The humanitarian and economic impact of the COVID-19 pandemic triggered the evolution of next-generation vaccine technologies. Some common approaches involve attenuated or inactivated virus, nucleic acid, DNA and RNA, and VLP, virus-like particle. We adopted the VLP approach. SARS-CoV-2 virus that causes the COVID-19 disease has 4 structural components. The spike protein S, a nucleocapsid protein N, the membrane protein M, and the envelope protein E. VLP-based vaccines typically have one structural protein that comes together as an ordered aggregated species and mimics the outer structure of the virus to elicit an antibody response. Since the SARS-CoV-2 virus uses its spike, S, protein to bind with the ACE2 receptor present on the surface of the host cell, any antibodies generated against the receptor binding domain of the spike protein will make the virus ineffective. This is why most vaccine candidates target the spike protein. However, there is a challenge. The spike protein is known to mutate, and when it does, it could reduce the efficacy of a vaccine that solely targets the S protein. Our vaccine candidate, DFEAT, targets not 1, but 3 structural proteins: spike, membrane, and envelope. Since membrane and envelope proteins are not known to mutate, this makes DFEAT, immunogenically, a better candidate for protection across emerging variations of SARS-CoV-2. Animal studies using the mouse models show that DFEAT is safe. And immunization with DFEAT induces SARS-CoV-2 specific neutralizing antibodies and antigen-specific humoral response. The titers of total anti-SARS-CoV-2 antibodies in mice closely match with those in convalescent patients' sera, demonstrating DFEAT's high immunogenic potential. When exposed to DFEAT, PBMC, cells from convalescent patients' blood show a T-cell response, which indicates that DFEAT mimics the SARS-CoV-2 virus and has the potential for long-term immunization. DFEAT has been developed using Decrypt, our proprietary Saccharomyces cerevisiae-based platform. Saccharomyces cerevisiae, or baker's yeast, is considered to be a safe model and has been used to develop vaccines for diseases like hepatitis B and HPV. Yeast is highly scalable, is expected to cost around 50% less than other technologies. We now prepare to take DFEAT to human trials.